Good morning, everybody. It's Friday. All right, well, let's get cracking. So, today we start really a large chunk of, well, what's rest of this section at least. And that very much leads on to what we're going to be doing next section, which is control of gene expression, right? So before we get to that point, we need to understand how gene expression actually works kind of mechanistically. And so that's what we're going to do over the next couple of weeks. And so this is really, I think this is actually one of the, I don't know, I like talking about inheritance. Inheritance is fun, but really it doesn't matter what you inherit, right, from your parents, if those genes or that information that you inherit doesn't get expressed, right? So really, um, you can't talk about inheritance, i.e. the traits which you inherit and the different types of phenotypes that you have without actually talking about how those genes are expressed and how those alleles affect that expression, right? And so really this is the basis of how we are what we are, right? And that information that we contains, thousands and thousands of different proteins that interact in different ways, expressed at different times, in different amounts, in different places. And it's really quite astonishing because if you think about it, every single one of your cells, apart from your red blood cells, have exactly the same genetic information, right? Exactly the same. Yeah, pretty much. Like mitochondrial DNA might vary a little bit, but that's always a funky one, to be honest. Um, so you have cells which are neurons, right? Which are like these tiny cells that have these enormous long processes, right? Literally, you have uh, neurons that have cell bodies in the small of your back, and their cells extend all the way down your legs to your toes. So next time you stub a toe, which I seem to do fairly frequently, remember that the reason why you can feel that is because you have cells in your body that are literally as long as your legs, right? And yet we also have cells in your stomach, right, which are able to tolerate really low pH and produce gastric acid, right? So your cells actually yourself, right? This isn't something that you kind of chug down and it stays there. This is something that you make. You actually make stuff acid, really strong acid, like pH. I think it was about pH 1.5, 2, something like that. Something that would do you an awful lot of harm if you came into contact with it, outside of your stomach. And yet, those two very, very different types of cells have exactly the same DNA, right? The exact same set of instructions. So they don't differ by the information they have, they differ because of the information they use, right? And that's both over kind of developmental time, right? So those cells that become neurons have a very different pattern of gene expression to the ones which become stomach cells, for example, right? But also once they are specified, once they are fully differentiated into that cell type, they also have very different expression profiles. To the extent which you can actually identify different cell types simply by what genes are turned on and which ones aren't in that particular cell, right? So how does all that work, right? So before we get to the kind of the detail on how those differences occur, we need to understand the basic process of gene expression. And that's really going from DNA through typically an RNA intermediate to protein. Also, sometimes just as an RNA, right? So it kind of very much varies um, depending on what gene is being expressed. And so this is a kind of a neat cartoon, sort of showing. I wish, in some ways, I wish I could find a picture which showed how busy and full this area is, because it kind of implies that there's this big open space where things float around in your cell, and that's very much not the case. But essentially, the general premise of gene expression, right, is that you have your DNA stored in the nucleus. You open it up to make it available for transcription. You copy that protein coding region, potentially, into an mRNA intermediate. And its purpose, by and large, is to take that information out into the cytoplasm. Because you can't make protein in the nucleus, right? You have to have that information out here, but you can't take DNA out of the nucleus either, right? So mRNA is the means by which you get that information out. And it's also the basis for the translation of that information into protein, right? Which happens once that mRNA has been exported and processed in eukaryotes. And we now get ribosomes assembling, which you see here, which bring in amino acids and make a polypeptide chain, which you can see here, right? So this is the whole process. What we're going to do is break it down into a series of, kind of bite-sized chunks, which are really transcription, the production of that mRNA transcript. In the case of eukaryotes, which are shown here because we have a nucleus, right? Bacteria don't have a nucleus. They don't have any organelles, actually. In eukaryotes, that mRNA has to be exported, right? And this is an active process. It's not like it just kind of falls out. It has to be directed and actively moved out of the nucleus. That mRNA also has to be processed, right? So in the original form which is produced, called pre-mRNA, that mRNA transcript cannot be translated. Actually, it'd be pretty catastrophic if it is. So in eukaryotes, that mRNA has to be processed before it's able to be translated by ribosomes. And so this is kind of what's shown here, this process where you have multiple ribosomes on a transcript translating that sequence information into polypeptide sequence. Okay, let's just get rid of this. I already know that I'm in my class because I'm in my class. Okay, so one thing, so you probably keep wondering why do I keep saying most of the stuff that does things is protein? Well, that's because there are also other active molecules that come from RNA, right? So we have well, obviously protein coding genes produce proteins, but there are also a whole range of other genes, many of which we don't fully understand, which produce RNAs which have a role in stuff. So obviously mRNA, that's produced by protein coding genes. We also have enzymes, right? These are called ribozymes. So ribosomal RNA is a ribozyme. Let's put, probably worth putting that in the chat. Come on. This is an RNA that acts, that is an enzyme. Right, so the ribosomal RNA is the active part of the ribosome. It's what creates the peptide bonds. We've got tRNA, short for transfer RNA, again used in translation, but this time it's a kind of structural intermediate that does the translating, right? That's actually what converts the genetic information in mRNA into the amino acid sequence information. And then we've got an entire universe, many of which I don't actually know, right? Because I just can't keep up. There's just so many of them, right? Of small RNAs that do stuff, right? So these RNAs often are involved in controlling gene expression, right? In different ways and in different places. There's actually, these are small nuclear RNAs. I don't know what SNO RNA stands for. I really don't. I should look it up. MI is micro RNA, but this is small nuclear RNAs. And so these actually get together with other proteins to help with RNA processing, mRNA processing. These form things called SNRPs, which I always think is pretty cool. I love saying that word. Anyway, so there's a whole panoply of genes which encode RNAs that do not get translated. These are called non-coding genes. They are still genes. Their expression is still controlled, but they do other stuff. So whenever I go, well, most of these things turn into proteins, well, that's what I'm talking about. Okay, and so as I mentioned with the first slide, there's really, you know, a series of steps, and one thing to keep in mind is the differences between prokaryotes and eukaryotes. There's actually some fairly fundamental differences between the two. The general overview is the same, but there's several wrinkles that bacteria utilize, which are very unique and serve a very particular purpose, particularly when we get to talk about the control of gene expression. So another thing that's really worth pointing out is directionality. And so you already know we have a double-stranded DNA, which has strands that go in opposite directions. This is important. And those strands have their own kind of directionality, right? But also that coding strand, let's say we're going to go from left to right here, because that's what everything else is doing. The coding strand encodes the information. I'll put this in there. Right, and so that's the top one. That's what we typically say is the gene, right? The template strand is used to make the mRNA, right? So this is the strand which is actually used by RNA polymerase to make a copy of the coding strand. And if you think about it, it makes sense, right? Because if you want the same base sequence, with some exceptions because of RNA, right? You can't use the coding strand as a template because that is that sequence. You need to base pair with the opposite strand, which is called the template strand. Right, so this is something to kind of keep in mind too. However, once you produce that mRNA, it has the same direction as the gene, right? It's the coding strand, which is the one at the top. And when that's translated into a polypeptide chain, or a protein eventually, that also has direction because it uses the mRNA in a very specific direction to be able to create it, right? So polypeptides or proteins have directionality too, right? Just like DNA and mRNA does. And here we denote it by, instead of a 5' and a 3', We use an N-terminus because of the amine group, which you'll see later when we talk about translation, and the C-terminus because of carboxyl group. And so polypeptides are always sequenced by adding new amino acids onto the C end, or the C-terminus. So that's also something that's worth kind of keeping in mind. However, the kind of the funky deal that this creates is that you have two potential coding strands. So you have one strand at the top going that away, and you have another strand at the top, bottom going that away, right? So you can actually encode information for genes on both strands and have genes which overlap, or like so, or even, you know, even more. It's rarer that they completely overlap because, you know, it'd be very hard for natural selection to select changes in one gene without them affecting another, basically. But you do get this situation where you can have, within a given stretch of DNA, multiple genes, and those genes are essentially encode their information in different directions because it depends on which strand is used to store that information. Does that make sense? Hopefully it does. So we have the top strand, right? So this encodes a gene kind of down here, essentially, right? And that template strand is used to make a transcript going in that direction because the template strand goes in the opposite direction. And on the bottom strand, this is a template for the gene or the information in this coding strand here, right? So this mRNA transcript gets made in that direction because that's the direction that the coding strand is going in. Template strand goes in the opposite direction. And sometimes you can have, and actually I've found these in my own work, you have genes which kind of go in opposite directions, but share a same kind of region between them, which contains regulatory elements, right? And so you can potentially control the expression of these two genes at the same time, which is pretty neat. Or even turn one off by turning the other one on and vice versa. So starting on the differences between prokaryotes and eukaryotes, one of the very big differences is that prokaryote genes are collinear with their encoding proteins. And so this might be like, well, yeah, of course they are, right? You know, the DNA produces mRNA, which produces protein. However, that applies for prokaryotes. It does not apply for eukaryotes, as you'll see in a second. So essentially, where you have your coding strand, this would be the one at the top. That's transcribed directly into mRNA, and the very same sequence is translated into amino acid, right? So wherever you have your gene, your mRNA lines up directly with that, your protein lines up directly with that, right? So that if you have a mutation in any given sequence, right, it corresponds to exactly the same position in that protein. Eukaryotic genes are not the same. They are far more complex. And so eukaryotic genes consist of a whole bunch of other bits and pieces, basically. So for a start, the mRNA produced is not collinear, or the final mRNA, right, which is the important point. So we have things called exons, and these exons are what encode the information that's going to be used to make protein. So these are the protein coding parts of the gene. However, these exons are separated by bits of DNA, sometimes pretty huge, in the thousands of base pairs, which encode nothing. They are just there, right? And they don't encode anything. They're not required for translation. In fact, you have to get rid of them before you can start translation. So the actual protein coding region of a eukaryote gene consists of a whole bunch of space which doesn't encode protein, right? That space being these introns. Also, prokaryote genes have what are called untranslated regions, or UTRs. They have one at the 5' end called the 5' UTR, funny enough, and at the 3' end as well. These are really, really important in the finished mRNA transcript for control of gene expression in general. RNA stability, which is part of control of gene expression, and also control of translation. Many of those non-coding RNAs that control gene expression actually bind to the 3' UTR. And finally, we also have what's called a poly-A signal, right? And this is used as part of mRNA processing to create a poly-A tail, right? A sequence of A's at the end of the finished transcript. So you can see there's an awful lot of stuff going on here, right? And the direction of transcription is the same. You get one large transcript, but that transcript has to be processed to get rid of these introns, right? And then obviously kicked out of the nucleus. Has anybody got any questions so far? Please do ask if you do. Okay, so obviously, as one could expect, all this empty space, and it really is by and large empty space as far as we can tell, right? There's nothing much there, right? And also these introns are highly variable, right? Because they are not protein coding, so there's no selective pressure. So these introns accumulate mutations without any selective pressure to purge them, from the population. Whereas protein coding regions obviously change much, much more slowly because typically, mutations will result in them not working. And so because of those introns, eukaryotic genes are huge, right? The actual protein coding region of genes, the exons, when you add them together and the size of the proteins themselves, aren't hugely different from prokaryotes, right? There are some real huge ones, but those typically result from lots of proteins getting together. But the actual size of the gene is way bigger, right? In bacteria, you're looking in the range of, 2 to maybe 6, 7 kilobases or 1,000 base pairs. And that's because they're organized in a kind of funky way, which they're not in eukaryotes. But in eukaryotes, it's about average of about 27 to 30 kilobases, right? That's huge. Okay, so let's get on to the actual nuts and bolts of transcription. So before we do that, there's a few things that are worth pointing out in terms of differences between DNA and RNA. The obvious one is that RNA has uracil instead of thymine. Ooh. I'm sorry. Uracil still base pairs with adenine, just the same way as thymine does, but it has a H there rather than a methyl group. Another really important difference, and this is actually part of why RNA is less stable than DNA, is that DNA is called DNA because it's deoxyribonucleic acid, right? And the deoxy is the lack of an oxygen here, right? Which if you look at any sugars like glucose, for example, every single carbon has a hydrogen and a hydroxyl group attached to it. So in DNA, that OH is swapped with an H instead. It's deoxy. RNA does not. So it's not deoxy, it's just ribonucleic acid. And so there's an OH here on the second carbon in RNA nucleotides. It's not actually used for, pardon me, it's not used for making mRNA, but it can actually be used for creating funky structures. And I actually might change this because I'm a little bit leery of saying it's can be. Oh, if I can spell can, come on. I think that's actually a better way of putting it, right? So DNA, unless it's being actively used by transcription or DNA replication, is always double-stranded, always. RNA can be single-stranded. It's not always single-stranded. In fact, RNA can form the craziest structures. Let's see if I can actually show you some. Get off. Come on. Go away. Go away. There we go. All right, so these are just some funky pictures, right? Of this is a tRNA would be my guess. Yes, so this is a tRNA, right? So this kind of shows you kind of the funky structures that they can form. They can base pair with themselves, so you actually get double-stranded RNA very frequently. Let's see if we can find any other. Oh no, here's another one, right? So this is a series of loops that are created, or these are called stem loops because they have a stem and then a loop, and then you can see how those kind of get bent and twisted around to create actual kind of three-dimensional structures, right? So anyone that tells you that RNA isn't interesting doesn't know what they're talking about, to be honest. Let's see if we can get some other pictures. All right, so this just kind of gives you the just general diversity, right, of funky ass shapes. They even call them crop circles. I don't know who dreamt that one up. Crop circle 2, so there must be crop circle 1 somewhere, right? So RNA can form all kinds of structures, and actually many of these structurs are really important in function, right? In their function as enzymes, potentially, their function as tRNAs for sure, and also it's used to, at least in probably more commonly in prokaryotes, to actually control mRNA transcription termination and also mRNA stability, right? So how long those mRNAs hang around for. So yeah, can be single-stranded. Doesn't have to be. Rarely, actually, to be honest. That's my little high horse. Actually, it's a very, very short high horse that I'm getting off of. Barely visible, but it's still worth pointing out. And this whole process is done by RNA polymerase, right? Again, we've talked about this before. RNA polymerase equals a hollow enzyme, right? Which is a group of proteins that come together or assemble to do a particular function, but they don't necessarily exist in that conformation all the time, right? So it's really like a it's almost like a group of individuals coming together to work on a project, and then they kind of go their separate ways when that's finished, right? Whereas a multi-subunit protein is a protein that's made up of lots of other proteins, but it always stays like that. So good examples are, like, for example, ATP synthase, right? I was reading up on that the other day. Is made up of, oh, I don't know how many, probably about 15 different proteins, give or take. But it always exists exactly in that format. You don't see it cycling between that and other forms. Anyway, so because RNA polymerase doesn't require a primer, it doesn't require a primer, right? And the same deal applies as with DNA, though, that nucleotides are only going to get added on to the 3' end of the growing strain, right? And so this is our DNA strand here. We've got our direction. We've got our T's, right? So you can know which one is which. But also going in this strand, we've now got that 3' OH, right? And we need that to be able to create a phosphodiester bond between that oxygen and this phosphate. Exactly the same as with DNA synthesis, right? It's not really, the principles are pretty much the same. Things that do it are different, obviously, but the principles are the same. And so RNA polymerase uses the energy of that phosphate bond here, right? Kind of splits off that 2 P's called pyrophosphate and creates a phosphodiester bond between that phosphate and that oxygen. Then it's incorporated, moves down another base, whatever the one is here, brings in its complementary RNA nucleotide, makes a bond, moves down, so on and so forth, right? You kind of get the picture. I'm really pleased, however, by this picture actually has the sugars in the right orientation. So that's really neat. You don't often see that. It kind of appeals to the pedant in me, I guess. And in prokaryotes, there's only one RNA polymerase holoenzyme. I don't know why that's in blue. But in eukaryotes, we have 3. Right, we have RNA polymerase 2, which is used for transcription of all the protein coding genes to produce mRNAs. So RNA polymerase 2 produces mRNAs, as well as small nuclear RNAs for SNRPs. Love saying that. RNA polymerase 1 has transcribes ribosomal RNA. So this is responsible for the RNAs that go into making ribosomes. And then RNA polymerase 3 makes tRNAs and one of the small components of ribosomal RNA. There's a larger one. I can't remember, 16S, I think, or maybe that's prokaryotes. I think 16S is the other one. S stands for Stromberg, believe it or not, which, if you're into old cars, is a type of carburetor. Isn't called it for that. It's because this is a, I think it's Stromberg's, or is it sedimentation? Pretty sure it's Stromberg's. It's basically how they sediment in sugar in an ultra centrifuge. That's all. Just in case you ever wondered. Probably didn't, but oh, you now know, right? Anyway, so RNA polymerase is, so this is number 2, same as RNA polymerase in prokaryotes. It does something really cool. So if you remember from DNA replication, you know, you get, you kind of pop open the DNA and then you have helicase and topoisomerase kind of extending that bubble, right? And so RNA polymerase does the same as those initiator proteins, right, that bind to the origin of replication. And then it bends the DNA, and in doing so, it pops it open. Now, that's a very transient single-stranded DNA situation, I guess, right? Compared with DNA replication, where it's single-strand and then it kind of gets, you know, replicated, and then it just stays apart in that bubble. So this is a moving bubble that will move along the coding region of the gene, right? And it'll be creating this little bubble as it goes, and as it passes on, once that mRNA has been transcribed, it will just seal back up again, right? Because it's not being bent out of shape, right? So the RNA polymerase physically twists the DNA and it pops it open, right? And that allows it to bring in RNA nucleotides or NTPs and add them using base pairing, right, with the template strand to add them to that growing mRNA molecule, which then kind of scooches off out of a hole, right? So the DNA comes in, kind of gets bent, exits up here, and in the process is transcribed into RNA. And so really, it's not that the DNA comes in, it's that RNA polymerase moves along, right? It's kind of a better way of thinking about it. So we've got a series of kind of steps, right? We've got we've got initiation, where we start the process. That's where most of the control of transcription occurs, by and large. We have elongation, which is just making it, right? And then we have termination, oops, put that back, which is when the whole process is finished. Because if you think about it, all along a chromosome, you've got hundreds or thousands of genes, right? You might only want to transcribe one of them. So if you just let that process go on, eventually it's just going to go all the way onto the end of the chromosome, and that'd be a huge waste, right? Because you'll be making stuff that you don't need and probably don't want, right? And so we've got to be able to stop this at some point, and that's a process which varies quite markedly between prokaryotes and even different prokaryotic genes and eukaryotes. So hopefully you remember when we were talking about origins of replication, right, in DNA replication, or replication origins even, how they have a higher proportion of A's and T's, right? So their AT percentage is higher. You find exactly the same thing in regions where transcription will occur, or where it will start. And so in bacteria, that's called a tartart box, or a -10 sequence, which sounds a little bit better. And -35 sequence, these are called consensus sequences, and we'll get to that in a bit. So this little red arrow always denotes where transcription will start. It's not necessarily the start of where translation will start, that's different. But this is where RNA polymerase will begin synthesizing the transcript. That's why this is called +1, because it's the first one. If you have 0, here's +1, that's -1 on the other side, essentially. Right, so the transcription start side is always +1. And this region is called a promoter. And so promoters are the part of the gene, and they are part of a gene, right? A very important part. It doesn't encode any protein, but is involved in the control of transcription. So in the very most basic sense, this is where RNA polymerase will bind, and transcription initiation factors will bind to begin transcription. In the broader sense, it's where anything that controls the transcription of that gene will bind, right? And so again, in prokaryotes, those regions tend to be relatively small. In eukaryotes, they are, wow, like huge, right? There are parts of the kind of the promoter which can be millions of base pairs away. I think they have to be on the same chromosome, I think. But I mean, I'm not entirely sure about that either. So these regions have to be recognized by the RNA polymerase and the transcription factors. Transcription factors and transcription initiation factors are proteins that recognize specific sequences of DNA, right? And those sequences are found in here. They don't necessarily all the same for all genes, they vary, right? But they recognize those specific DNA sequences, right? And so these are the sequences that are typically recognized by RNA polymerase and transcription initiation factors, sigma factor, I think, in eukaryotes, right? And these are consensus sequences because at these distances, so -10 and -35, you have something which looks more or less, right, like this sequence in bold at the top. Doesn't have to be exactly the same. So for example, down here, we've got, what's that, REC-A, so that's a recombination protein involved in DNA repair, I think. This is a complete copy, right? So that's a perfect -10 sequence. Look at the -35 sequence and it differs here, right? Others are less close, right? And so what we mean by consensus sequence is what is the base found most often at that position, right? And so if you look through here, T is the base found most often because you have a C here, but the rest are T's. Always A, mostly T, right? And so you can actually visually express that in one of these kind of bit plots. So for example, these two positions here, you only ever find A and G. That's it, right? This one here, you almost always find T, but sometimes you find C and whatever that little bit thing squished underneath it is, right? And so from this, you would probably say, well, this is a TTCAG, CACC, meh, meh, GG, ACAG, meh, 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 C consensus, right? Where you can't really make a call for these because, you're pretty even distribution of bases, at least between two bases at that position. So this is what's called a consensus sequence. And also how closely the actual sequence at the -10 and -35 boxes match that consensus sequence has consequences in terms of how strongly RNA polymerase and transcription factors will bind this sequence, right? So we can have varying strengths of promoters. And really what we mean by that is how much expression we're likely to get from that gene, from that promoter. So the higher the promoter strength, more frequently RNA polymerase will bind, more frequently that gene will be expressed. And so overall, you'll expect a higher level of expression from that promoter or of that gene that is controlled by that promoter. Flip side also applies where if there's a relatively weak affinity, so that consensus sequence has diverged and isn't really that close anymore, then RNA polymerase will be much less likely to bind and stay bound to that promoter. And so overall, you'll get lower levels of expression or transcription, at least, from a gene controlled by that promoter. So obviously, over time, mutations can change those consensus sequences, right? So now you can start seeing at like a DNA level how mutation can start changing function of, or even function maybe, the output of different genes, right? Start creating novelty. Does anybody have any questions so far? Let's check the chat. Yep. All right. Okay, so initiation in bacteria is fairly straightforward. Actually, it's pretty straightforward. So we have our promoter, typically relatively small. This is our transcription start site. Our sigma factor, which is a transcription initiation factor in bacteria, uh, kind of comes in with RNA polymerase, and then you're off at the races. That's it, pretty much. Right. So the RNA polymerase holoenzyme and the sigma factor will process along. It kind of goes ding 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 like funicular railway. It's a cool word, funicular. It pops open the double strand bit of DNA at the promoter, right? And so again, lots of T's and A's, right? Even some T's and A's over here, right? So these are particularly the -10 sequence. We'll see a similar thing in eukaryotes. These are very much easier to open. So not only does RNA polymerase bind here, but it also uses that to kind of pop open the double strand to be able to add the RNA nucleotides to the template strand. Now, one thing to remember is that different to DNA replication, this mRNA transcript doesn't stay bound to the template strand, right? So it's not like DNA replication where you have your template strand and your newly synthesized strand, right? Basically stays stuck, and then you have double-stranded DNA that's new. In transcription, that it will start and it will basically kind of end up as you go along, kind of tailing off the bottom, right? So that allows, once the RNA polymerase has gone along, for this bubble to close back up, right? So it becomes double-stranded DNA again. Now, obviously, once that RNA polymerase has kind of got out of the way, another RNA polymerase can come in and repeat the same process again, right? It's not you have to wait till you're finished. You just have to wait until it's moved on, right? Okay, so I think we'll leave it there for today. Right, I want to get on to eukaryotes next week. I think it's a good place to stop. Does anybody have any questions about this or anything else? Yep, got 3 minutes, peeps. No, oh no, no questions. Don't forget, you're going to be grilled like pretty solidly on this stuff in the exam. Okay, so make sure you understand kind of all these different bits and pieces, because there are a lot of them, right? What things do which, and also the general principles involved, right? So if I keep banging on about something like, hey, TA rich sequence in DNA, that's going to be where the DNA is going to be split apart, right? You can be fairly sure that something alluding to that will be on the exam. All right. Yeah, so what I actually recommend for studying these kinds of processes is actually to do like make your own cartoons, right? Like storyboards, I guess. Draw stuff out. So just looking at this, it's not really going to go in very far and stay there. But if you actually try and draw out, make your own like little kind of mini poster of transcription, let's say, or translation, or whatever, right, that we're working with, then that stuff sticks better, right? And also, if you try and do that without the use of your notes or these PowerPoints, you'll soon figure out what bits you remember and which bits you don't, because the bits you don't remember, you won't remember, right? So that's a good way of finding out what you don't know. Cool. All right, everybody. Have a fabulous weekend and uh, rest, relax, study hard, the usual, right? And I will see you on Monday. See you, everybody.